you know about all the low-tech stuff. Now we come a little bit into the present with some of the modern tools used in biotechnology. And the first thing is, like, I'm not even going to really talk a lot about getting DNA out of cells because it's so easy. You basically need soap and salt and alcohol. Soap to break up cell membranes. Salt to get the proteins away from the DNA. DNA is kind of wound around proteins. Salt kind of helps that unravel, and then alcohol gets. You basically need a place where alcohol meets water. You know how they kind of layer because one's less dense and one's more dense. DNA likes to go to that interface between the alcohol and the water, and it's kind of as simple as that. It's pretty easy to get DNA out of cells. But once you do, what you've got is a bunch of cells worth of DNA, which is a bunch of, if it's a human, it's a bunch of 6 foot long, all globbed up globs of DNA, but it's hard to really do anything. So the first thing you need is scissors. The first thing you need is a way of cutting the DNA into more manageable pieces. And that is where what we call restriction enzymes come in. You know what enzymes are. These are particular enzymes. And there are literally hundreds you can buy. That have various differences. They're all fairly expensive. But you could buy them. It's a little intimidating to buy and use restriction enzymes because you spend like a couple $100 and what comes in the mail is a styrofoam box packed with dry ice and like one of these. Just a little microcentrifuge tube with like maybe half a centimeter of fluid in it. And that's what you bought for $250. What it does, if you put it in solution with a bunch of DNA, is those little restriction enzymes glom onto the DNA and start kind of spinning along that DNA all through it, looking for one specific DNA sequence. And every place they find that sequence, they cut. I'll show you the biggest company that sells them. They're called New England Biolabs. See if I can find a price for you. Okay, so this is just the A's, for example. These are all different restriction enzymes. Pick one I recognize here. Eco 1. All right. Eco R1 tells you what the sequence is it cuts at. It looks for GAATTC. Let's get a price. So we're looking at, it's 20,000 units per mil. This is a low concentration, costs you $62. If you want the high concentration, it's $254. You know about how much a milliliter is, volume-wise. It's one of the small ticks on the thing. Well, you're ordering 50,000 units, and it's 100,000 units per milliliter. So you're getting a half a milliliter for $250. It's intimidating. Better use it right? But yeah, these are available. And so each of them looks for a different sequence. I'm going to give you two examples, two of the older, more well-known restriction enzymes. And I'm only giving you these examples because of one important difference between them. You'll never have to memorize the sequence they look for. I'll always tell you that. So for example, this one's called HIND2. They are named just as weirdly as genes, don't worry about it. But HIND2 cuts anywhere it finds GTCGAC. And when it cuts, the way it cuts is straight through the double strand between the C and the G, and the G and the C. Notice I give you the recognition sequence GTCGAC, but when I show you the cut, I'm using the double strand because that's how DNA is, right? It's double stranded. And what you get, every place it cuts, is fragments which have what we call blunt ends, because this enzyme cuts straight through the double strand. Which is fine, depending on what you're looking to do. If you want to do DNA fingerprinting, where we identify someone based on their DNA, an enzyme like this is fine. If we're looking to find a gene just to study it, not to stick it in some other cell, an enzyme like this is fine. However, if, as you are, you're looking to do something a little crazier, if you got your eyes on the prize, if you're shooting for glow-in-the-dark cat, you're going to need a different kind of enzyme. You're going to need an enzyme like EcoR1, the one I just priced for you. EcoR1 looks for GAATTC. Again, don't memorize that. Not worth the brain space. What's important about it is the way that it cuts. It cuts in a different way than HIND2 does. It cuts in a zigzag pattern. Okay? It cuts like this, between the G and the A here and the G and the A here. And when the pieces come apart, they have what we call sticky ends. Keep in mind, when I draw this, this is a much longer double-stranded DNA. So we're looking at like, and then on this side, there was a whole bunch more this way, right? But these are just the ends of the pieces you've cut. And this right here is where the money is, because those sticky ends is what you know about nitrogenous bases and weak hydrogen bonds. You know that it kind of doesn't matter, DNA, RNA, whatever. If the hydrogen bonds will form, you can stick two pieces together. A pairs with T, G pairs with C in DNA. Because look at what it leaves. It's TTAA on one side and AATT on the other side, basically that means those two pieces, if you let them, they'll bind right back together. That's not the important part, though. The important part is any two pieces that you cut with EcoR1 will recombine like that. So if restriction enzymes are the scissors, EcoR1 is both scissors and glue, right? Any two pieces cut with the same sticky ends type restriction enzyme like EcoR1 will recombine with each other. That's what we mean when we say recombinant DNA. Now you start to see the possibilities. Now you start to see, oh, we can take, say, the gene that makes a firefly's butt light up. We play it right, we can cut that gene out with EcoR1, stick it in another animal's cell, cut that animal's DNA open with EcoR1, and if you're lucky, you can get the piece that has the gene for the light up butt to fit right in there and reseal, right? And the sticky ends will match right up. And now the firefly butt gene is in another animal's genome. Let me show you a simple example.
A common technique in engineering with to a loop.
All right, that's not good. Okay, so there's EcoR1 clamping onto a bacterial loop of DNA. It's spinning on the DNA, looking for what it's looking for. When it finds AATTC, it cuts. As long as the other piece has been cut with the same sticky restriction enzyme, it'll glom right onto that sucker, and now you've got some other gene in a bacterium. Now, what this is referring to, the reason it's showing you this with a bacterium, is that the very first use of this technology was by a company called Genentech, and what they did was they cut the gene for human insulin out of a human cell. You know, insulin, right? The stuff that type 1 diabetics have to inject themselves with. We used to get it from like crushed up cow pancreases, very expensive. They went ahead and snipped out that gene from a human cell, did exactly what I just showed you into a bacterial cell. And suddenly they had a bacterium that would make human insulin. Why is that useful? Well, because bacteria are way easier to raise than cows. And so all you need now is a vat full of like bacterial nutrients. And that bacterium will do the thing that bacteria do where they make more and more and more exponentially. And suddenly you got a vat that's pumping out gallons of human insulin per day. And you have a multi-billion-dollar corporation now called Genentech, right? So that was the beginning. We've come a long way since then. Anyway, that's tool number one I want to show you today. Tool number two, equally important, because once you cut the DNA, you're not done. I kind of fast-forwarded with the Genentech stuff and the glow-in-the-dark cat, because what you need before you can do any of that is a way to find the pieces you're looking for. And that's where gel electrophoresis comes in. Gel electrophoresis is a way to sort your different size pieces of DNA by size. And what it uses to do that is essentially almost the same stuff as Jell-O, believe it or not. It's a setup that looks very high-tech and really isn't. It's, in fact, this setup right here you're looking at. All you need is an electrical power supply. What's essentially a tub that you fill full of salt water with a lid that has electrodes on it that will electrify the salt water. Okay. And into that tub, you put a flat block of Jello. Not exactly Jello. It's called agarose. It's actually made from like algae, I think. It's fairly expensive, but I found this stuff from Thailand. It's actually food, but it's called agar powder. And it seems to work about as well. So we might give it a shot at some point this week. Anyway, you do it just like you're making jelly. You boil up the powder, and then you pour it into, this is a fairly small one, but you just put these bumpers, and it gives you this area where you can, sort of a shallow little bathtub you can pour the Jello into, right into here. And before it hardens, you put what's called the comb in there. And the comb is just something that makes a bunch of little indentations in the Jello for you to insert your mixed fragments of DNA. Are you with me so far? What you're going to get is a flat block of Jello with holes in it. Okay? So you pour the gel, and then you insert your cut up pieces of DNA into the wells that you've made with the comb. That's a pretty good diagram of, here's the tub, here's the flat gel, here are the little holes that the comb created. You insert you take a little micro pipette thing and put your samples in here, and then you turn on the power supply. And the thing is, assuming you've set up your negative and positive electrodes correctly, DNA is a little bit negative. DNA is a little bit negatively charged. So you just make sure which one's negative and which one's positive. The universal kind of color code. Which one's positive? Red. Red is positive. So you got to make sure that your samples, which is where the comb is. Oh, when the gel's done, you pull the bumpers off. And you put this sucker right in here, and you make sure that the sort of the foot of the gel is on the positive side, and that's going to pull your DNA through the gel because DNA is a little bit negative. And you're thinking, why does this matter? Who cares? Well, pulling that DNA through the gel it's harder for the big pieces to get through the Jello than it is for the little pieces to get through the Jello. So the little pieces move faster. So they're essentially sorting themselves out by size for you. Now, you can't see the DNA pieces as they move because DNA is clear and very small. So what you do once you're done, and I'll tell you how you know when you're done in a minute, what you do once you're done is you use a special stain that'll make the DNA visible. The only thing you have to really worry about with gel electrophoresis, it's not a super complicated process, is timing. You really got to worry about timing, because if you run the electric current too long, all those pieces will go through the gel and out into the salt water, and you lose them. You want to make sure to run it just the right amount so that it sorts all the pieces by size, but not so long that the smallest piece runs out of the gel. And the way you fix that, the way you make sure that happens, is you also insert some dye, some colored dye that you know moves faster than the smallest pieces of DNA. And so as soon as the dye makes it to the end of the gel, you stop, you turn off the juice, right? And what you're left with is a pattern that looks something like this, right? The biggest pieces won't have moved that far from the origin, what we call the origin of them, right? Because they were so big that it took them this long to even just get that far. The smallest pieces will have moved the farthest. Now, if your question right now is, if they're different size pieces, why are all those bars the same size? That's an excellent question. They're not the size of the pieces, they're the size of the wells in the first place. What's in each of those bars is, hundreds or thousands of that same size piece, all moving together. But they move faster because they're smaller, or slower because they're bigger. How are we doing? This is kind of the big one for the day. This is the one that takes some figuring out. So I've got an activity for you to do. Any questions? Once you've done this, and you've got all these pieces sorted out, it's not that hard to find ways to actually pull pieces out if you know which pieces you like, which pieces you want. And there are ways to make those pieces show up on a gel different from the others, okay? So this is the beginning of glow-in-the-dark cat and bill, right? It's the scissors and the glue and the sorting machine. I've got an interactive kind of online lab about electrophoresis for you I want you to work on. It's not hard, but it really does kind of take you through the basics.